So the cell cycle. The way that I remember the cell cycle is like this. I remember I prefer monkeys at the circus. And you can interpret that whatever way you would like. That I prefer when there's monkeys at the circus, or I prefer my monkeys at the circus. Like when I go to the zoo and I see monkeys, that's off-putting. I'd rather see them at the circus. Right? I prefer monkeys at the circus. All right. Anyways, so where does this come from? Why do we say it? It's because these are the phases of the cell cycle. I is for interphase. P is for prophase. M is for metaphase. A is for anaphase. T is for telophase. And C is for cytokinesis. Okay. Now, of these phases here, prefer monkeys at the, or prophase, metaphase, anaphase, and telophase, those are all part of what's called the M phase, right? So these parts here are part of M phase. Okay, and that would lead you to believe that M phase is very long because it's got 4 individual phases in it, but it just happens to be that lots of things are happening there, and so we say that we have an individual phase for each one of them. The vast majority of the cell cycle, about 90% of it, is going to occur in interphase, right? So if we were going to draw a pie chart, okay, here's a pie chart. This pie chart is going to have all the different parts in it. If I was going to draw all the parts, I would say that this part here, that's about M phase right there. And maybe even a little smaller than that. M phase. Okay? And then the rest of it is going to be interphase. Okay? The whole rest of it is interphase. But interphase gets divided into a couple different parts. Okay? First of all, there is a very important part at the very beginning of interphase that is called the G1 phase. Okay? And G1 phase is a growth phase. It's the time right after the cell divides, and so this cell is going to have to grow a little bit bigger in order to get back to its normal size. And it's also the time where the cell is going to really do whatever it's supposed to be doing. It's going to be doing all of its work, right? And so then you've got this spot right after G1 phase, or between G1 and the next phase, right about here. Okay, and this spot is the G1 checkpoint. Okay, the G1 checkpoint is a time when the cell is going to check for all the important things. It's going to check for cell adhesion. Meaning, is this cell attached to the extracellular matrix? Are we around other cells that we're supposed to be around? Okay? It's also going to check for cell density. Is there enough room around this cell for us to divide again? Okay? Or is this going to be a crowding situation? Okay? Tumors, cancerous tumors, obviously do not go through and check for cell density, because if they did, they would notice that there's way too many cells of that type around, and they're in much too close of a confined area, and so they would never divide. But one of the things that cancerous cells do is they tend to ignore G1 checkpoint, right? It also checks for DNA damage. Has the DNA been messed up at all in any way from any kind of thing? And if it passes all those things, this generally means that the body might need more cells. Okay? Is there a need for more cells? If there is a need for more cells, then they can go into this next phase, which is called the S phase. And the S phase is when the DNA is going to get copied. Okay, that's when the actual chromosome or chromatin rather in this case gets a second copy of it, so that there'd be 200% of the amount of DNA in the cell after this S phase. And then after S phase, there's another sort of little checkpoint, right? And then it moves on into G2 phase. Okay, and during G2 phase, there's another regulation point, another spot here. Okay, that's called G2 checkpoint. And G2 checkpoint checks for the last final little bit to say, hey, if we're going to go ahead and divide, we're going to make 2 more cells here. Let's make sure we did everything correctly. Okay, so a lot of the problems that are going to happen in G2 checkpoint are errors in DNA replication. Okay, and it's going to check to see, okay, well, we went through S phase. Did everything get replicated properly? If so, that's fine. We can move on. If not, we have to figure out, first of all, is it possible to fix the errors? And if it's not possible to fix the errors, that cell then goes into what's called programmed cell death or apoptosis, right? So if you fail G2, you either get repaired or destroyed. Okay? If you fail G1, you either get destroyed or you can go into a spot called G0 phase. Okay? So after G1 phase, you can sometimes go into a phase that is called G0 phase. Okay? And in G0 phase, it is like a pause button. The cell cycle is paused. Okay? It can be unpaused if the situation is correct, but it will not move on until something happens to make it move on. Okay? Your muscle cells are constantly in this G1 phase. So if you don't damage your muscles, if there's no lactic acid buildup that causes your muscle cells to get hurt, if your muscle cells don't tear because you're doing, some strenuous activity, then your muscle cells don't need to divide. And it takes a lot of energy to make new muscle cells, and so it's actually good from an evolutionary standpoint to have them not be constantly dividing, right? But if you want to get big muscles, one of the ways that you do that is you exercise, right? And if you want to bulk up, anybody know if you want to just not be super strong, but you want to just look giant, how do you do that? Besides steroids. Anybody ever learn that? No? Well, one of the ways that you can do it is low reps, high weight, right? So the whole point behind low reps, high weight is to damage your muscles, to tear them, right? Because when you tear your muscles, they're going to have to make new muscle cells. And when you make new muscle cells, you get bigger, right? But if you do high reps, low weight, you're just getting lactic acid buildup, and that's going to cause your muscles to be conditioned. It's going to cause muscles to be repaired, but they're going to just be the same size or relatively same size. And so if you're doing high amounts of reps and low amounts of weight, then you're likely to get in shape. You're likely to have toned muscles. But if you're doing low reps, high weight, that's going to make you look bigger because of the way that it's tearing the muscles and creating scar tissue and also creating new muscle cells, right? There's your weight training lesson for the day. You can obviously tell that I really am a subscriber in the bulk up. Anyways. Interphase. So we said that this whole segment right around here, everything in this part, this is all interphase, right? And I haven't drawn it perfectly, but interphase should represent approximately 90% of the cell cycle. Represents about 90% of the cell cycle as far as time goes. Okay, so 90% of a cell's life will be spent in interphase. Sometimes much longer than that. So muscle cells will be much longer in interphase because they're going to go through a longer period of time before they go into M phase. Some cells will never go through M phase. So like nerve cells, some nerve cells won't divide ever And so those will spend, 99.99999999999% of their life in interphase because they had to divide one time when they were a little baby non-differentiated cell. So let's talk about what happens in interphase. First of all, we say that interphase is really the time in which the cell is doing whatever its job is, doing its cellular work. Okay, so cells perform their specific tasks while in interphase. Okay, so let's take a specific cell. Let's talk about, let's say, a pancreatic cell. Okay, so pancreatic cells, one of the major jobs of the pancreas is to produce all of the digestive enzymes, right? So you've got like your lipases, those are the enzymes that break down lipids. You get your proteases, those are the enzymes that break down proteins. And then you've got your like amylase and stuff like that, the ones that are going to break down carbohydrates, okay? Those are produced in the pancreas. And so in a given day, a pancreatic cell's job is to just get its endoplasmic reticulum rolling, the rough ER, and start producing all these enzymes and then export them into this thing called the common bile duct, and then the common bile duct moves them into your intestines where digestion can happen, okay? So, in order to make all those enzymes, they've got to have the instructions to make those enzymes, and the instructions to make the enzymes are contained in the DNA in the cell, okay? So we know that these pancreatic cells, they have to have access to the DNA so that they can know how to make all the enzymes that they're supposed to make so that they can do their cellular job, right? Now, the interesting thing about this is that since all cells are the same, the thing that really determines whether or not it's a pancreatic cell or a skin cell, because you know that your skin cells from day to day are not producing digestive enzymes, we're not digesting everything that we touch, that would be weird. So why do pancreatic cells make those enzymes, but skin cells do not? And that all comes from the fact that in a skin cell, parts of the DNA are clumped together. They're clumped together so much that nothing can access them. The parts, the enzymes that would go in and create RNA can't get into that spot in the DNA, and so they don't make whatever that DNA says to make. Okay? So if you clump up the part that has all the digestive enzymes in it, that means that that cell will not produce digestive enzymes. But if you take the part that has digestive enzymes and you spread it out, that means that that cell can then produce digestive enzymes. That's the whole idea, is just clumping together or spreading them out. So if the cells are going to be doing their job during interphase, that means that the DNA has to be available during interphase, meaning that it's not going to be in the form that we constantly think about DNA, and that is in chromosome form. When people think about DNA, they're like, oh, it's in the chromosomes. But we'll learn that that's really not correct. Okay? So we say that the cells must have access to their DNA. And therefore, DNA must be in chromatin form, which is spread out. Okay? And this is in contrast to chromosome form. Chromosome forms are all clumped together. All the DNA gets clumped together then. Okay? So let's talk about the types of interphase, or the parts of interphase. The first part is called G1 phase. It's part of interphase. G in this case stands for growth. It's a growth phase. And if you look at our little pie chart, what's happening is you're going right from M phase, where the cell's dividing, into G1 phase, right? And so at this point, the cell has just divided, and so it's going to have to recover from the fact that it just divided. We can say that the cell recovers from cell division And it's going to do that by growing larger. The cell becomes larger. And it's going to do that by replacing the organelles so that there's the correct amount of organelles. So organelles must be replicated. Okay, so after these two things happen, the cell is ready to go. The cell is at its tip-top shape. Okay, it's ready to perform its function the best that it will ever be able to in its life. Okay, so after these two things occur here, the cells are most functional directly after G1 phase. Okay, so if you were going to arrest a cell or stop the cell cycle or pause the cell cycle, the time that it makes the most sense to do that is right after G1, because that's the time where the cell is the most efficient because it's the correct size and it's got the correct amount of organelles. Okay, so G0 phase can pause the cell cycle after G1. Okay, and the reason why that might happen is that something happened in G1 checkpoint to say, hey, we don't need any more cells. Stop making cells, and so then it just doesn't, and it won't make cells then until. It won't make cells then until it gets the signals, the growth signals that say, hey, go ahead and make more cells. Okay? If it doesn't get the go-ahead to make more cells, though, it will go into what's called S phase, which is another part of interphase. S phase is synthesis phase. And we say that the DNA is replicated to make a second copy. Okay? So you start out with this double-stranded DNA, and then you're going to end up with two identical strands of double-stranded DNA after S phase is complete. Okay? The reason why it's called synthesis phase is because when you make new DNA, that's called DNA synthesis. So producing DNA is called DNA synthesis. Then you go to G2 phase. Now, this is slightly disingenuous because G2 and S sort of overlap sometimes. But we're going to consider them as sort of separate parts. Still part of interphase here. This is another growth phase. We know that because it's G, right? This growth phase is one where the cell is going to prepare to divide again. So the cell grows in order to get ready to divide. So the cell becomes larger. Slightly larger. But more importantly, extra copies of important organelles like mitochondria are produced. Okay, and it has to do this because it's going to split into two cells, and each of the cells then is going to have half as many organelles as it used to. And so if you have one where you already don't have that many of them, like mitochondria, you need to make more of them so that each of the cells will have enough to be able to do its job. Okay. After that, you've got M phase. M phase is not part of interphase. It's the rest of those things up there. Prefer monkeys at the. Okay, and there's two types of M phase that we're going to look at in here. One of them is called mitosis. And mitosis is a single parent cell producing two genetically identical daughter cells. Okay, and this will produce what's called somatic cells. Okay, and somatic cells are pretty much just like normal cells. So most of the cells in your body are somatic cells, like skin cells and liver cells and pancreatic cells and stuff like that. Those are all somatic cells. Okay, the other one is called meiosis. And that's a single parent cell. Produces 4 genetically unique cells with half as many chromosomes. Okay, these cells that have half as many chromosomes, if they started out as, like in a human case, we start out with 2 copies of the chromosomes, right? After they have half as many, they're going to be called haploid. We go from diploid to haploid, and this produces sex cells in animals, which are called gametes. Which are sex cells which are sperm and egg. And the last thing, cytokinesis. That happens during telophase. So this is the actual splitting or actual division of the cell that occurs in telophase. So at the end of telophase, still in telophase technically, cytokinesis begins, and then telophase ends when cytokinesis completes. Okay? I will post your reading guide online for you guys to print out.